substance, substantia grigia centralis. The gray substance consists of two symmetrical portions, one in each half of the medulla spinalis. These are joined across the middle line by a transverse commissure of gray substance, through which runs a minute canal, the central canal, just visible to the naked eye. In a transverse section, each half of the gray substance is shaped like a comma or crescent, the concavity of which is directed laterally, and these, together with the intervening gray commissure, present the appearance of the letter H. An imaginary coronal plane through the central canal serves to divide each crescent into an anterior or ventral and a posterior or dorsal column. The anterior column, columna anterior, anterior cornu, directed forward, is broad and of a rounded or quadrangular shape. Its posterior part is termed the base and its anterior part the head, but these are not differentiated from each other by any well-defined constriction. It is separated from the surface of the medulla spinalis by a layer of white substance which is traversed by the bundles of the anterior nerve roots. In the thoracic region, the posterior lateral part of the anterior column projects lateralwards as a triangular field, which is named the lateral column. Columna lateralis, the lateral cornu. The posterior column, columna posterior, posterior cornu, is long and slender and is directed backwards and lateralwards. It reaches almost as far as the posterior lateral sulcus, from which it is separated by a thin layer of white substance, the tract of Lissauer. It consists of a base, directly continuous with the base of the anterior horn, and a neck or slightly constricted portion, which is succeeded by an oval or fusiform area termed the head, of which the apex approaches the posterior lateral sulcus. The apex is capped by a V-shape or crescentic mass of translucent gelatinous neuroglia, termed the substantia gelatinosa of Rolando, which contains both neuroglial cells and small nerve cells. Between the anterior and posterior columns, the gray substance extends as a series of processes into the lateral funiculus to form a network called the formatio reticularis. The quantity of gray substance, as well as the form which it presents on transverse section, varies markedly at different levels. In the thoracic region, it is small, not only in amount, but relatively to the surrounding white substance. In the cervical and lumbar enlargements, it is greatly increased. In the latter, and especially in the conus medullaris, its proportion to the white substance is greatest. In the cervical region, its posterior column is comparatively narrow, while its anterior is broad and expanded. In the thoracic region, both columns are attenuated, and the lateral column is evident. In the conus medullaris, the gray substance assumes the form of two oval masses, one in each half of the cord, connected together by a broad gray commissure. The central canal, canalis centralis, runs throughout the entire length of medulla spinalis. The portion of gray substance in front of the canal is named the anterior gray commissure, that behind it, the posterior gray commissure. The former is thin and is in contact anteriorly with the anterior white commissure. It contains a couple of longitudinal veins, one on either side of the middle line. The posterior gray commissure reaches from the central canal to the posterior median septum and is thinnest in the thoracic region and thickest in the conus medullaris. The central canal is continued upward through the lower part of the medulla oblongata and opens into the fourth ventricle of the brain. Below, it reaches for a short distance into the filum terminale. In the lower part of the conus medullaris, it exhibits a fusiform dilatation, the terminal ventricle. This has a vertical measurement of from 8 to 10 millimeters, is triangular on cross-section, with its base directed forward, and tends to undergo obliteration after the age of 40 years. Throughout the cervical and thoracic regions, the central canal is situated in the anterior third of the medulla spinalis. In the lumbar enlargement, it is near the middle, and in the conus medullaris, it approaches the posterior surface. It is filled with cerebrospinal fluid and lined by ciliated columnar epithelium, outside of which is an encircling band of gelatinous substance, the substantia gelatinosa centralis. This gelatinous substance consists mainly of neuroglia, but contains a few nerve cells and fibers. It is traversed by processes from the deep ends of the columnar ciliated cells which line the central canal. End of section 5.